Alrighty, thank you everyone for waiting. We had a little technical difficulty, but we are set. So thank you for joining us tonight for the Prevention and Wellness series, live from the Center for Hope and Healing at Cancer Survivors Park. Cancer Survivors Park is designed to serve as a gathering space for multiple organizations to share their messages of health and healing. The park also serves as a focal point for education about cancer prevention, community events, and inspiration for changing the way we think about cancer. The gardens, pavilion area, and healing spaces are gathering grounds for patients and caregivers to gain perspective and strength, and for communities to celebrate life. The park is located in Greenville at 52 Cleveland Street, just off the Swamp Rabbit Trail. Before we get started, I'd like to introduce myself. My name is Melissa Aguilar, and I am the Community Outreach Coordinator for the Center for Integrative Oncology and Survivorship, and I will be your host tonight. Tonight, we'll be joining our regular host, Dr. Jeffery Giguere, as he will guide us in conversation about the science of epigenetics. Given the growing number of lifestyle-associated cancers, it is becoming increasingly clear that early molecular events that may increase the risk of a future cancer could be arrested or reversed via lifestyle modification and early intervention. Tonight, we'll examine how the human genome is played over the course of a lifetime and why your DNA is not your destiny. We will also learn more about the Prevent Cancer Greenville research study, a first of its kind research study that aims to further our understanding of how one may reduce their chances of developing cancer from preventable causes. Viewers are encouraged to ask questions in the chat box throughout tonight's presentation, and I will be sure to read aloud all questions as Dr. Giguere will provide best possible answers. And with that, Dr. Giguere, take it away.
Thanks, Melissa, and I appreciate everybody's patience. That was entirely doctor produced as far as being cancer or computer illiterate, but my IT support here has helped me out, so hopefully we can get through this and still finish up on time. So if you've been tuning in for the Cancer Prevention and Wellness series, you know we've talked about lifestyle and lifestyle medicine, and how you can improve things from the standpoint of your diet. And we've emphasized a plant-based whole food diet. We've emphasized exercise. We've emphasized diminishing our stress. We're going to, in future months, talk about the power of nature, and other things from the standpoint of improving the environmental factors. There's also things you can do to alter your environmental factors to the negative. It's important that you realize that while you get DNA from your parents, it is not necessarily a situation, and now this is not advancing. Let's see. Honestly, guys, we practiced this and it worked fine. Here? Okay, all right. Another. So here's the central dogma that a lot of us that were part of science in the 70s learned, and that was that we inherited our DNA from our parents, and we got 23 chromosomes from mom and 23 chromosomes from dad, and that was pretty much set. And these were made up of units called nucleotides, and together they made up base pairs, and they were translated or expressed via a transcription to messenger RNA. Now, all of you are scientists when it comes to messenger RNA, from the standpoint that you realize that the vaccines that became available to us in November, the initial one, Pfizer, and then Moderna, As you remember, these were messenger RNAs. They were messenger RNAs from the spike protein of the virus. And these messenger RNAs within the body would then translate into proteins that would establish an immune reaction to declare us immune. But what we have learned in the last 20 years is that what happens between expressing the DNA into protein can change based on intragenerational factors of the environment. So what is the code within the genome or within the DNA that you get from your parents is not necessarily what becomes your destiny. So the genotype does not become your phenotype. What happens and what, so for example, you come from a tall mother and a tall father, and you have genes that imply that you should be tall, but perhaps your social environment is that you have some food insecurity or some other things that affect how tall you can become. And so while your genotype says tall, your phenotype says less tall. So there's lots of factors, including DNA penetrance, to determine how the gene is interpreted into real life. So this is a busy slide, and it basically just tells you kind of the breathtaking speed in which genetics has changed. I was in undergrad in the 70s, and none of this existed. So Frederick Sanger developed a method for sequencing DNA, and people developed DNA sequencing machines. And in 1990, the NIH took on a significant project looking at a cell's DNA and actually mapping every base pair, every nucleotide base pair within a human genome. So this started in 1990. It finished 2 years early in 2003, so 13 years. We went from defining 1,000 base pairs a day to where we are now defining 1,000 base pairs per second. This project took 13 years. As I mentioned, it took $3 billion. We can do a next generation sequencing of our DNA now in about 10 days, and it costs $600. So what we're going to see in our lifetime is that we are going to have our genomes as part of our medical records because there's going to be pharmacogenomics as to how we're going to react to drugs, and can we determine that we're allergic, and how are those drugs going to be metabolized. So we've gone further. We can now determine what genes cause what diseases, what genes can potentially cause certain cancers. In the second decade of this century, Doctors Emmanuel Carpentier and Dr. Jennifer Dudick, both from France and from the United States, developed the CRISPR technique, which stands for a long word, which I won't bother you with, but this is allowing us to do outside of the body gene editing. And we're actually looking at that from the standpoint of certain diseases that are basically caused by one gene that we've delineated, and we're starting to do that with sickle cell in the hematopoietic stem cell, as far as taking the bad cell out and repopulating it, but taking the bad gene out and repopulating it with a good gene.
So, Dr. Giguere, when you talk about gene editing, do we have to worry about how ethical the science is? It's crazy to see how fast all these advances and discoveries seem to have taken place over the last 20 years.
No, you're exactly right. Genetics has always been haunted by the ethical component. I always talk about the fact that our science gets ahead of our ability to know how to use it. But in genetics, we've clearly misused it. There were a number of studies with regard to twins in the concentration camps with the Nazis. We practiced eugenics in this country with trying to sterilize the folks that were perhaps had some genetic defect and perhaps create some kind of eugenic product. But when we talk about genomes being part of our health record, I mean, we've seen all these cyber attacks and whatnot. I mean, there's lots of things that come up, but when we start looking at the possibility of playing God, we have to figure out what the rules are going to be. I remember when Dolly the sheep got cloned, and was it going to be a situation that we were going to be able to clone certain people, and would that be a situation where the rich would get cloned and the less and the minorities would not? So there's clearly within genetics ethical questions. I suspect that that's going to be as big a field of science as genetics is, and that is how correctly to use it. So, from the standpoint of genetics, we went through Gregor Mendel and his P experiments and how things were inherited and autosomal dominant and autosomal recessive. Darwin talked about evolution and the survival of the fittest. So these were all DNA changes, but they happened over many, many generations to figure out whether something in the DNA actually was going to provide a person with a survival advantage. And then, in the early part of the 20th century, we got the concept of the gene, and the concept that the gene could be mutated. And this genotype not necessarily being the same of what we got when we got everything put together in the phenotype, what we looked like. We might have been, destined to be brown-eyed, but we ended up being blue-eyed. And then in our era, we saw Watson and Crick and the double helix and how DNA and what it was made up of and how it got actually translated and expressed into proteins which made up organisms. And in the 21st century, we're seeing some of the effects of what we call epigenetics, which is another layer over top of the DNA and sometimes affecting the messenger RNA to alter the way that genes are played per se. So that's going to be the focus of our lecture going forward. So epigenetics refers to those processes that drive gene expression. The DNA sequence is housed within that gene, but how it gets expressed can be changed based on this extra layer of interpretation of what genes get expressed and what genes don't. For example, in the embryo, we all get the same DNA. Every cell gets the same DNA, but there are things that are affecting that DNA so that a skin cell becomes a skin cell and a liver cell based on the environment becomes a liver cell. And they all have different functions and different activities based on the environment and how those genes are expressed. So the epigenetic biomarkers are inducible. If the environment changes for the better or for the worse, they're reversible. They are cell specific. They can be affected by smoking. So if you start smoking, it may affect the epigenetic markers. If you stop smoking, it may affect them for the better. Same thing with alcohol consumption. Physical activity, couch potato versus triathlon. Things change from the standpoint of which genes get expressed. Diet's a big part of that. Environmental pollutants, so we'll talk more about those things. There's some things that are endogenous factors like getting older and the cells become more senescent and oxidative stresses happen and they're less likely to be dealt with effectively, and certain metabolic and hormonal disorders can all have an effect on how the gene is expressed.
I have another question. So what about the social environment? How does that social factors come into play with all of this?
So let's say you live in a neighborhood where there is nutritional uncertainty and economic uncertainty, and where are you going to be living that night, and is there violence in the neighborhood, and so all of that affects people's stress, and so even though you may be given a great gene profile from your parents, the environment in which you grow up and the adverse childhood experiences and that sort of thing can certainly have an effect on how the genotype becomes phenotype. And if you get into a better environment or you find your way out of the bad environment you're in, those things are reversible. So yeah, great question. So the father of epigenetics was a British doctor by the name of Conrad Waddington, and he talked about the fact that there was a branch of biology that could affect the genes and how they were actually expressed. And so this is his landscape, epigenetic landscape. And so up here at the top, you have this pluripotential stem cell, and as it goes through gravity, it can go down these various valleys. And this landscape, based on what you do lifestyle-wise, what your environment is like, that sort of thing, this landscape is malleable, and it can be changed over time, so that cells can be altered, and the way the DNA is expressed can be altered as well. So the genetics is the genome, and that's the DNA, and certainly it can change over time. There's mutations that are happening in the DNA all the time. Usually they cause the cell to die. Sometimes they can be genes that can turn an oncogenic or cancer promoter on, or it can turn a cancer suppressor off, and so that may turn into a malignant cell. But the alterations in the DNA that happen based on environmental influences are secondary to epigenetics. So this is my metaphor for the genome. The genome is the piano, and the epigenome is playing the piano. And you can play some keys, and those become evident. They're notes. Some keys you don't play, and they're maybe suppressed. You can play chopsticks, or you can play Beethoven symphony in C minor, and some notes are played over and over again, and some notes are repressed or depressed. So it's all on how you play the piano, and our objective here is to try to get you to play the piano the best you can. All right, so this is probably the most scientific, and I thought probably the title of my talk might keep some of you away, but I see a lot of people are on there, and I congratulate you on this. But the DNA is wrapped in these nucleosomes, which together is called chromatin. And so if you go down here to this box, and I'm going to try to get my cursor to go down here to this box, but so there's a couple of turns of DNA around these protein molecules called histones. Some DNA is more open, and it can be methylated. It can be marked with DNA methylation. Methylation is a very common organic chemical. Methyl, or the acetyl group, is a carbon atom and 3 hydrogen atoms. But the methylation can also occur at this histone tail, and so it can unwrap the DNA and expose it to the potential of being transcribed into RNA. If it's DNA that shouldn't be exposed, then bad things happen. If it's a DNA that would be better exposed and transcribed, better things happen. There can also be post-transcriptional changes. So the non-coding RNA can happen to the messenger RNA so that it doesn't become a protein. And We had a branch of oncology called missense or nonsense RNA that really never took off. But there's a number of factors: methylation of the DNA, methylation of the histone or acetylation of the histones, and this post-transcriptional micro mRNA or non-coding RNA, microRNA. So this is epigenetic modulation that gets to the actual formation of a cancer. You can have methylation that either suppresses the inactivation of parts of the genome, or it potentially promotes an oncogene or a cancer part of the genome. So cancers can be formed by epigenetic changes in the DNA, and also changes in how post-transcriptional RNA is done. So there's a lot of things that affect the epigenome that are out there in the environment. So out in Seneca, where I worked for about 10 years, there's the Oconee nuclear plant for Duke Power, and they have chemists there, and these chemists are exposed to benzene and the metabolic byproducts of benzene. And that can alter the genome with hypermethylation so that you are more predisposed to leukemias and to multiple myeloma. So Duke Power would send their chemists over every year to be evaluated. I don't think they're necessarily exposed to benzene as much as they were, and they've certainly done mitigating. They've mitigated the environment where there's too much exposure, but we would look at them every year to determine if there were any early effects of the benzene on the genome. The uranium is something that can be something that is held in the granite in our soil. So it's something that we have to be aware of, especially if we're drinking well water in the upstate. Overall, it's implied that too much methylation, too much acetylation, too much methyl marking of the DNA is a bad thing. And you are better off to have less methylation. So this was a study that looked at the probability of survival based on the number of genes that were methylated. And you can see that in this survival plot, what we call a Kaplan-Meier plot, the cancers that had greater than 50 genes methylated had a survival, a median survival close to 6 to 12 months, while the patients who had less than 50 genes methylated had a median survival that really hadn't been reached yet, but was out, over 2 years. So we've talked in terms of the intra-generation during the person's life changes that happen with epigenetics. Are there environmental factors that can influence the genes that can actually stay within a genome so that they're actually placed in the sperm or the egg for transgenerational impact? So there was a study that was done at Överkalix, Sweden, which is a small area in northeast Sweden, which in the turn of the 19th century was pretty much cut off from the rest of society. And so they were very dependent on whether their crop was a good harvest or not. And so over the course of generations, scientists looked at these patients who had parents that lived in periods when they were prepubertal, lived in periods where there was famine, and lived in periods when there was more feast. And the progeny of patients who lived, who had parents who lived during their early years in famine settings had a longer life expectancy. The people who lived, who had progeny and lived when they were young in famine situations, feast situations, had a shorter lifespan. So much so that the difference between the famine parents and the feast parents ended up overall to be a 32-year life expectancy difference. There's other things from the standpoint of parents who had most of their calories generated from meat versus vegetables, and cigarette smokers, which produced sons and grandsons that were more obese. So this could happen over not just one generation, it could happen to their sons and it could happen to their grandchildren and their children and their grandchildren. Also, there's this circumstance where at the end of World War II in 1944, the Germans stopped the ability for the Netherlands to get much in the way of transported food. So it was called the Dutch Hunger Winter, and mothers who were, ladies who were pregnant at that time of famine had an increase in children who were obese, diabetic, and there was also neuropsychiatric consequences. And this occurred up to 3 subsequent generations of those affected. So
I wanted to comment, it's interesting that these 2 instances had 2 separate outcomes, but nevertheless affected future generations.
yeah, well, one was prepubertal and one was where the women were actually pregnant with a child. So the child was getting, less than 1,000 calories per day. So there may have been different physiologic situations going on.
Yeah.
But sometimes these markers are so intense that they actually they transfer over generations. That's usually occurring in those genes that are more in charge of metabolism and sometimes more in charge of psychiatric issues.
It makes you think about what you eat, how it might affect your future children and grandchildren.
Or if you can eat.
Yeah.
So in the social environment, are you getting enough to eat, and is that the right kind of food?
Right.
So, there's clearly differences that we can hopefully discover as we look more intensely at the DNA and the changes in the DNA, the so-called genome, the changes in the epigenetics, the epigenome, the transcriptome, what the RNA looks like between healthy people and cancer people. We're going to probably see some markers, some biomarkers that are going to help us determine who might be more inclined to get a malignancy and potentially do something that intervenes in that regard and can produce some prevention. So that's a lot of what's going on now from a genomic standpoint. We're doing our own project in Greenville, which we're going to talk about. There is a more national project called US2. So a lot of work going on in that regard to try to see if we can find within this genetic capability that we are now developing. And the epigenetics are not well understood, but there is an epigenome, and we can certainly look at that, and we can look at that year to year to see how that changes based on changes in our environment. So it would stand to reason also that if the epigenome is having an effect on whether cancers develop or not, that there would be some therapeutic agents that would be developed by the pharmaceutical companies. So we have these histone deacetylase inhibitors that are available, FDA approved. We use these. We don't use them very often because they are really dedicated for diseases that we don't see that often, although the panobinostat is now approved for later therapeutic lines of therapy for multiple myeloma. We also have been very familiar, and it's been very active for a number of years, to have hypomethylating agents, so DNA methyltransferase that decreases the amount of methylation that occurred in the DNA. And these have a therapeutic effect in myelodysplastic syndrome as well as in AML. So it stands to reason that there's going to be a lot of places where the epigenome can potentially be influenced by drugs, and most of the pharmaceutical companies always looking for another way of defeating what is a hard to defeat malignancy. Will look for other methods. And I just have a quote here that successful approval of the first generation of drugs intended to target epigenomic epigenetic pathways, has convinced almost every major drug company to invest in cancer epigenetics. So more to come from the standpoint of therapeutic capabilities.
The good news is there's things that we can do to help our epigenetic outcome.
Well, we're going to talk about that. Now, this is where we're going now. So cancer prevention and wellness. Wish that every doctor had a compartment in their brain that was dealing with cancer prevention and wellness. But in the world, on our planet, especially in America, we are more reactive than proactive. And we are more inclined to treat disease rather than to prevent disease. And there's lots of reasons for that. And we're going to kind of go over those reasons, but here's the breadth of the problem. And you can see that from 2008, 2016 to 2020, this number of cancer cases, this number of cancer deaths, this amount of money that we pay because we have this disease process within our midst. This is despite the fact that the death rate is decreasing. We're getting better with regard to being sensitive in our surveillance and picking up cancers at a point where they're still potentially curable. And we are also getting better with regard to our therapeutic interventions. But we're not getting better at cancer prevention. And you can see last year, 630,000 cancer deaths. I mean, we just went through a pandemic, and to date, that hasn't caused that many deaths in the United States as cancer did. Although, over 1 year, a virus doing that kind of damage is still significant. So, that's the significance of the problem. You can see that when you look at these layers, that the real, you can't see it all the way maybe, but this is like from 55 to 64. And so all these are occurring, these layers are getting bigger as we get older, as our cells become more senescent. The process of cell division increases the likelihood that there's DNA damage and that there's potentially a driver mutation for a cancer to develop.
So we spend a lot of time focusing on best practices for cancer survivors at the Center for Integrative Oncology and Survivorship. Where are the survivors on this graph? And when we look at 2040, what does that mean?
So, if you look down here at this number, hopefully you can see my cursor. We've gone from 3.6 million with the estimate in 2040. We're going to be at 26.1 million. Obviously, I'm sitting here at the Cancer Survivors Park. We're going to have a lot more survivors. Some of them have been effectively treated, but they've been treated with medicine that could potentially cause other problems down the road. There's always the risk that their cancer could come back. There's always the risk that genetically a second malignancy could happen. So it has been the situation historically that when we get to an arbitrary 5-year survival and there's no evidence of disease, we give them back to their primary care doctor. And one of the things that is occurring within the Center for Integrative Oncology and Survivorship is that we have this lifetime clinic. This is a clinic where we are really focused on what the patient received, what the kind of cancer they had, what was the histological grade, what's their likelihood of recurrence, what do we need to continue to monitor them well without causing too much in the way of scans and which cost money and produce radiation issues. And we also want to make the point that along with the personal history, the thing that changes that may actually turn into something that makes us think of a syndrome is the family history. So we do a better job than the treating oncologists of following up on what's happened in your family to see whether there's anything there that we need to do from a genetic standpoint to see if there's any kind of a germline mutation. As far as challenges to cancer prevention, which is where we certainly have to go, there's really just too many silos. You get your colonoscopy here, you get your mammogram here, you get your pap smear here. We have lung cancer screening now for a certain population that has been exposed to too much smoking. Cancer genetics is coming along, but it's still kind of in its infancy. But the major thing is that there's been no hospital driven prevention mindset. There's been no American Medical Association prevention mindset. And more importantly, there's been no insurance, Medicare, CMS mindset that says prevention. So we don't get reimbursed for it. But it's the way that we lower the cost of medicine in this country and in every country. And we just have to make a commitment to it going forward. So that's my soapbox. Another complicated slide. Basically, this is a pie chart that shows all the things, and if you can read all these things, most of them, the enemy is us. So it's tobacco, it's obesity, it's lack of activity. Obviously, you can't necessarily affect the viruses you're exposed to. You can't necessarily affect the environment, but you certainly can affect a lot of these things. And these yellow boxes are basically there to tell you what you can do to live a healthier lifestyle so that your epigenetics are happy, so your epigenome is happy, and that your DNA is being translated and played like that piano in a better way. But there's also a situation where the word has to get out to our marginalized population, our minority population, so that they understand how important it is to live a lifestyle that has a better likelihood of keeping you cancer and chronic disease, and keeping chronic disease from happening, like diabetes and hypertension and whatnot. So there's a lot of things, but we know the enemy, and the enemy is us. So I think, and Dr. Gluck is on the, this is one of her slides, and it's a money slide. So if you really talk about the genome here, the genome is the encoded part that you get from your parents. It may change with mutations, but it's transcribed into these messenger RNAs, which we know about because we have a virus that has we have a vaccine that has a messenger RNA. Those translate into proteins that build organisms which are infected by the outside environment, whether the social environment, whether you're exposed to toxins, whether you're exposed to cigarette smoke or alcohol. And this environment affects your epigenome that regulates the genes, and round it goes. The only thing that's really malleable in here is your environment, because you could fix that. And the way that you fix that so that the genes are better is that the epigenetics are also malleable. So fix your environment, the epigenetics will change as well within a generation. Oh, boy, we almost got to the end, and now I'm having trouble advancing the slides again. Let's see, hit every button possible. Okay. All right, so this is a project that was developed by Dr. Larry Gluck as well as Leanne Perkins, who's a nurse practitioner who did an internship at the NIH in cancer prevention and genetics. And so what we're doing is with this project, we're seeing patients longitudinally year over year, and not only are we looking at them and telling them about lifestyle and changes that they can make, but we're also looking at their weight, and we're also checking their body composition, and we're also placing blood samples and and urine samples and buccal swabs into a biorepository so that we can look at how their lives have changed and how their genome and epigenome and transcriptome and proteome and metabolome are all changing along with it. This is the wave of the future, and it's a huge project, all supported by philanthropy. So we talk about their exposures and how they might lighten those exposures to be better exposures than noxious exposures. We do their body composition, and then we do these biomarker testing that we will look at getting objective information from in the future. This is the device that we use, a SOZO device. You basically get on here with bare feet and no metal. Put your hands on the top, and this evaluates your water, your muscle mass, your fat mass. And so if you're competing against yourself year to year, which we would expect you to do, we would expect that these numbers would get better as well. You may also find that when you go to your internist, when you go to your primary practice doctor, that your cholesterol goes down and your blood pressure goes down, and you get a lot of potential positive reinforcement from the advice that you're getting via this program.
Yeah, I wanted to comment on the program. It's such a cool study that's 100% free, and aside from checking to see where you stand in terms of cancer prevention, it's also kind of like a free yearly checkup to see what your body mass is, your hydration levels. You learn a lot about yourself.
Yeah, you go in there with no expectations and you leave with none, but you come out and say, okay, if they can measure all this objectively, then I can certainly get behind changing my lifestyle, because it's just one of those things where your doctor's saying, hey, you look good, and a lot of other things that are objectively telling you that you're doing better is a whole lot different.
Right.
So here are your visits. This is a cell that potentially has a DNA molecule that's gone bad and is potentially going to become a cancer cell someday. And over time, it's doubling. And so this cell that's got an oncogenic potential is now 2 cells, and now it's 4 cells, and now it's 8 cells. And it gets to a point where it's big enough where it can actually have the property of metastasizing and thus becoming incurable. So we're looking at you year over year, and we're testing these biospecimens, and we're keeping those in the biorepository to see if there's any markers that can tell us, this is where he is or she is now. And we know that that's going to lead to something, and maybe we can intervene at that point. So every year you have this Center for Cancer Prevention and Wellness visit. This is called Prevent Cancer, the Greenville Project. You do all these things, the body composition analysis that we talked about, and you get a lot of advice, and then you get blood drawn and a buccal swab and urine, and that all goes in the biorepository. So this is how you get involved with it. I know, like everything else, the people that are on here for cancer prevention and wellness are usually Caucasian women. We have probably almost 600 people that are participating in this now, and the vast majority of them are Caucasian women. So if the Caucasian women could ask their husbands or significant others, so we need more male participants, and we definitely need to be totally looking at the entire population and having it fit the demographics of the upstate. We need more minority participation. Again, every one of these is free. You have to show up. It's not anything we can do virtually. But there is a website there, as well as a phone number that you can enter into the program. So we
And when you
encourage.
say free, there's no insurance involved, nothing, 100% free, correct?
Yeah, no, it would take a whole nother full-time employee to work on insurance here. So yeah, we didn't want to get into that. And Dr. Gluck and his team have done a great job of getting enough money to pay for all of this as well. So all good. So we talk about fruits and vegetables. We talk about stuff coming from the ground. And some of these things actually work via the epigenome. And so you look on here and there's some histone modification things, and there's some DNA methylation activity here. And I'm presuming that these are hypomethylating agents, and that the histones that make up the chromatin are acting and behaving better because of your ingestion of these fruits and vegetables. So it's important that we say that lifestyle medicine is something that you need to practice. I mean, we certainly, and I for many years have given targeted therapy in the form of hormonal manipulations and chemotherapy, and we've done radiation therapy, and we've done surgery, and all of that has been reactive, but we're not going to make a huge dent in the incidence of cancers or chronic disease without lifestyle modification. And we live in America where our lifestyle is probably the worst. So if you can just go a year without taking a right or left into a fast food restaurant, that's a start. And then for optimal success in preventing and controlling cancer, once diagnosed, we can also make a difference by behavior risk reduction. So hopefully they can do things to make their treatment go better and perhaps more successful. If the treatment is successful, maybe we change lifestyle to keep the disease from coming back. And even in a situation where the disease is eventually going to cause your demise, you can live longer and better with your lifestyle choices. So let's go back to that area where we're seeing these patients year to year, and we see the difference in their weight, and they see the difference in how they feel, and we see the difference in their body composition. And eventually, based on all these changes, we're going to see a difference in their genome and their epigenome and all those other things that we are taking samples from now to see how they match up. So with that, I know I rushed through that. We are finishing at 6:30, but we didn't start till 5:40 because I'm an idiot with a computer. But if there's anything in the chat box, then I'm happy to go there. I can't hear you, Melissa.
Does anybody have any questions for this evening? We did not have any questions in the chat box yet, so everyone is being pretty...
Or they're asleep.
They're blown away by all the information. So Wendy said, how do we get involved in the program? We can put this information in the chat box for the Prevent Cancer Greenville study, but all you have to do is call the number and we can get you scheduled for an appointment.
And I should mention, the program is run by Julia Yates and Susan Webb and Armand Desailliers, and they are just superb clinicians. And so it's a very, very comfortable appointment.
Yes. And to answer the second question, can anyone join even if they haven't had cancer? Yes.
Oh, great question. I'm sorry I didn't. Anybody 18 or over, they can't have active cancer, but they can have had cancer. But no, we want healthy people. So this is not for people who had cancer. It'd be great for people who maybe feel like in their family they've had a lot of cancer and they may be predisposed. And we would love to see those people, but we need more male representation. We need more minority and more marginalized population too. I think they also have to speak English.
Yes, it doesn't need to be your first language, just need to speak English.
Right.
We have a few more questions. Can identical twins be affected differently in utero by epigenetics?
The correct answer for me on that is I don't know. You know, they do have the same DNA. We don't see that the epigenetic influences from one generation transfer to the next generation very often. They have to be under the extreme circumstances that we talked about with the Dutch hunger winter and that sort of thing. So I would think not, but I don't know that that's the case. Certainly, twins living in a different environment would be affected from their environment via epigenetics differently. But epigenetics is more of an environmental thing than a thing that you get from inheriting your genes.
What else? Do we have a CRISPR in our community?
No, CRISPR is still very investigational. I can't imagine how expensive it is. There's not going to be a whole lot of these around. So this is very, very experimental still. And then, you know, like we talked about, not only we have that technology, but we have to figure out how to correctly use it.
And then we had another question about the participation in CCPW, the prevention study. And the question is, even people who have diseases like diabetes.
Oh, yes. Yeah.
Yeah.
Other chronic diseases, you know, we probably want to follow you for about 10 years. Again, I'd like you to be around for 10 years. So, yeah, no, type 2 diabetes, love that. Type 1 diabetes, insulin dependent diabetes that's well controlled, be fine.
Yes. I think that is all the questions we have.
And that's great. So I think people need to get on with their evening and dinner. And again, this is also recorded. So you can see all my ahs and whatnot again. But if you felt like it was worthwhile and you feel like somebody else would get some benefit out of it, just let them know that in about 24 to 48 hours, it'll be on our website here at the Cancer Survivors Park. So that's Cancer Survivors Park, all one word, .org.
Yes. And I just put the phone number in the chat box to call to schedule an appointment for the study. And I will just quickly go through and let everybody know about our next month's discussion on breast cancer prevention. This will be presented by Dr. Brian McKinley and of the Prisma Health Breast Health Center Breast Health Center at Prisma Health on Thursday, July 8th from 5:30 to 6:30 p.m. And studies have shown 1 in 8 women in the United States will develop breast cancer in her lifetime. Join us as we discuss key risk factors, screening guidelines, lifestyle modifications to reduce your breast cancer risks, and breast cancer survivorship. And that is all for tonight. So thank you.
You're going to tell them with a quote.
Oh, and yes, I have a quote in the custom of our closing statement that I will leave you with something short and sweet. The best time to plant a tree was 20 years ago. The second best time is now.
All right, so let's get on that, guys. Thanks for your attention.
Thanks, everyone.